So by now we have been talking about this particular chapter, that is biological classification, and we are talking about the different kingdoms, and we have completed the kingdom Monera, we have completed kingdom Protista, right? And we have also completed the kingdom Fungi, also known as kingdom Mycota. In these, we have talked about the general characteristics, we have talked about the mode of nutrition, the habitat, and certain classes that they are further classified into. For example, in fungi, we talked about the certain classes. What can anybody tell me what are the different classes we talked about in fungi? Anyone?
Sir, phycomycetes.
Very good. Phycomycetes, then
Ascomycetes.
very good, and B Basidio
And
Basidomycetes
and last
and
one. Very
Deuteromycetes.
good. So these are the different.
Deuteromycetes.
Very good, very good. So these are the different classes that we have learned in the fungi. Now the next kingdom that we have in line is going to be the kingdom Plantae, and the last one is going to be kingdom Animalia. Right. So we have already discussed these three, but we are going to discuss in detail about the kingdom Plantae and Animalia. As I have told earlier, we are going to have separate chapter on this. But the basic idea you have to understand is, here plants, you have to understand that most of them are autotrophic in nature. Right. So these are autotrophs. Whereas Animalia are heterotrophs. Please switch over your microphones. So these are autotrophs and these are heterotrophs. Now they have the chlorophyll or the chloroplast, and that will enable them to make the food for themselves. Whereas this Animalia, they actually depend on the autotrophs or the Plantae for their food requirements, or some other organisms, as they're dependent on others. Now there is another mode, that is the partial heterotrophs, we can call it. Okay, please switch off your microphones. Okay. Anyway, so here there is a mode called partial heterotrophs, or that is known as the parasitic mode of nutrition in case of plant. Now you might ask, so plants are typically autotrophs, then how you are saying parasitic? Now there are certain cases, for example the pitcher plant, or the let's say Venus fly, one second, I'm sorry, Venus fly trap. Okay, now in these Venus fly trap, what happens is the plant is going to get partial nutrition from the insects. So what they do, they trap the insect, okay, and then they're going to derive nutrition, a particular type of nutrition, out of this trapped insects. And typically what happens, these insectivores plants, so these are called what do you call, insectivore plants, right? And these insectivore plants, typically they can grow in nitrogen deprived or nitrogen deficient soil. Okay, so what I'm saying is, these insectivore plants like the pitcher plant or the Venus fly trap, they can grow in a nitrogen deprived or nitrogen deficient soil. So to cope up with the nitrogen, or the requirement of the nitrogen here comes from this trapped insect. So this trapped insect, when they are going to be digested and going to be absorbed, they are going to get the nitrogen out of here. So the plant, insectivore plant, gets partial nutrition in the form of a parasitic mode, or in the form of a partial heterotrophic mode. But when we talk about the Animalia, we all know that Animalia is going to be completely what? They're all heterotrophs in nature. They are dependent on the autotrophs or other organisms based on their food habits. Like for example, they are going to have the herbivores, carnivores, and omnivores, right? Based on that, they are going to have the different food requirements. Now these are basic differences, but apart from this, there are many other differences, right? There are many other basic criteria, basic feature on which we have divided the Plantae and Animalia into further groups. So we will be talking about all of those in the further classes in the next chapters. But right now, let's understand a very important concepts regarding virus, viroids, prions, and the lichens. Now, why these are important is because these organisms or these living, they are all living organisms, but these are acellular organisms, right? And very important thing, they are not placed in any of the groups, any of the kingdom that we are talking about. So they are not placed in any of the 5 kingdom that we have talked about, and that's why they're important. They're a little bit unique, they're a little bit different than the rest that we have talked about. So let us understand one by one in this particular session. So first, let us start with the virus. So virus, you have to understand, they are neither living or non-living. So they are, you can say that neither living nor living, or you can say they are both living and non-living. It is all dependent on where they are. So if they are inside a host organism, right? What is a host organism? It's a living organism where the virus can, let's say, enter. So if there is a host organism and the virus is going to gain entry into the host organism, till the time it is inside the host, it is going to be living. And the moment it is going to be outside the host, right, it is going to be non-living. So the living and non-living criteria, the living and non-living situation of the virus depends on where it is. Is it inside or is it outside? So the host, the position of the virus inside or outside of the host decides where it is going to be. Now, the virus again are non-cellular organisms, and they're going to have an inert crystalline structure outside the living cell. As I said, they're dead. So inert means they are non-reactive, they cannot react. So they are non-reactive, non-living structures where when they are outside the living organism, and they become alive, or they are actually living inside a host organism. So they hijack, right? I use the word hijack because the moment they're getting into the host, right? So you have the virus, and this virus, once it is in the host, it is going to use the host machinery to make more of virus. So multiple copies of virus is going to be made once the virus gain entry to the host, and it is going to use its own mechanism, the host mechanism, to make copies of themselves. So one virus enters the cell and it is going to take over the mechanism of the cell, all the biochemical mechanism, and then it will make multiple copies of the same virus. So in that way, viruses are very unique. In that way, viruses are very clever, I would say, because they have a very less amount of genome. The amount of genetic material is very less, but they are able to hijack and use somebody, like a parasite, right? It is not having it on its own. It is going and taking over of the host machinery, and that's why they are also referred to as the obligate parasite. Now, there are a few scientists' names that you have to understand. The name virus was proposed by Dmitry Ivanovsky, and his use of the term because of the nature, that is venom or poisonous fluid, and he recognized this. He saw the virus for the first time on a microbe that is causing the mosaic disease in the tobacco plant, tobacco mosaic we call it, and that we today know as the tobacco mosaic virus, TMV. Now, the Benedict, what he did, he extracted the fluid from the infected tobacco plant or the TMV, the plant affected by the TMV, and he then transferred it to the healthy plants. And he found that these healthy plants, when we are giving them this infected tobacco plants extract, it is actually causing the healthy plants to become infected. And that's why he actually suggested the term contagium vivium fluidum, that means infectious living fluid. Now, Stanley, on the other hand, he crystallized the entire virus, and what he found is the virus here is mostly, or the crystal here is mostly formed of proteins. Akshita. Right. So when we talk about the viral structure, you have to understand with the help of, let's say, understand this with the help of this particular adenovirus, a simple structure here. Now, what happens here is we are going to have this capsid. The outer layer is going to be called the capsid. This particular part is called the capsid. And this proteinaceous capsid layer is actually formed of capsomere. The individual units here, you can see these individual units, these are called the capsomere. And there are going to be certain glycoproteins which are also present, and on the center you are going to have the DNA or the genetic. Here we are talking about the genetic material. Okay, we are going to have the genetic material in the center surrounded by this protein layer called the capsid. Now we have some other, for example, you are going to have the HIV virus, in which you have additional proteinous layer outside, that is a virus envelope. But again, we don't have to worry about those structures in detail. Also, we are going to have the bacteriophage. What are bacteriophage? These are the virus infecting, or virus that infects bacteria, right? So the virus that infects the bacteria is called bacteriophage, and you can see here you are going to have this head structure, then here you are going to have the sheath, and you are going to have the tail fibers here. So there is another picture which will be seen later of the viral structure. Now when we talk about the virus, as we said, there is going to be a protein called capsid, and there is going to be a subunit. Each of the subunit is going to be called capsomere, and this capsomere can be arranged in a helical or polyhedral geometric form. Different types of the arrangement of the capsomere is there depending on the type of organism. And the role of this capsid or capsomere is to provide protection to the nucleic acid. And the genetic material, right, in this case of virus, can be DNA or can be RNA. It depends on the virus type. Now the plant viruses typically have ssRNA. SS means, here it is called single stranded RNA, right? That is called ssRNA. Now when we talk about the animal virus, it can be ssRNA or dsRNA or dsDNA. DS means double stranded. Okay, SS refers to single stranded, DS refers to double stranded. Now when we talk about the bacterial virus, that we are talking about the bacteriophage, you can see there is going to be a double stranded DNA. Now viruses are pathogens. They cause disease in organisms, animals, humans, plants. Like in our case, we have the mumps, smallpox, herpes, influenza. We also have the AIDS. AIDS stands for acquired immunodeficiency
Deficiency syndrome.
Deficiency syndrome.
syndrome, right? It is not a disease, it's called a syndrome because it's a combination of symptoms, right? And people die because of the lack of immunity that is developed when the people are suffering from HIV AIDS. Obviously it is caused by the HIV virus or human immunodeficiency virus. So in case of plants, the symptoms can be the mosaic formation, the damaged leaves, the leaf rolling, curling, yellowing of the leaves, right? Veins, damage to the veins of the leaves, dwarfing, short height of the plant, and stunted growth, lack of the proper growth. So these are some of the symptoms that we see in case of plants and the diseases that we see in case of humans. Now, a very important thing that is a little bit extra we have to understand is how actually, what are the different life cycle of a virus. So to understand that, let's understand a particular phase. So we have a phase, and this phase DNA, it is going to attach to the host cell, right? So because it is a bacteriophage, this is going to be a bacteria. So the bacteria can be, let's say, E. coli for simplicity. So in the bacteria, on the surface of the bacteria, the phage DNA comes and attach, and it is going to release the DNA or the genetic material. So in one type of life cycle, what I call Lytic cycle, the bacteria is going to be injected with the phase DNA, and the phase DNA, it is going to, right, take over the host machinery, right, and it is going to produce new phase DNA and proteins, and they are going to be assembled into new phase. Phase here again we are talking about new virus here, right? So we create new virus in the Lytic cycle, and that new virus is finally, when you have enough of the virus, finally what is going to happen is it is going to lyse. Lyse means it is going to destroy the cell, right? It is going to destroy the host cell and finally release the phase or release the viruses, as you can see. And the viruses are then free and they can infect another host cell or another healthy cell. So here in the Lytic cycle, the host cell is dead, new phase are or new viruses are created. Now let's look at the second cycle called Lysogenic cycle. In the Lysogenic cycle, it is slightly different than the first one. Here what happens is, as the virus enters or the DNA enters, the phase DNA or the viral DNA enters the host cell, it is going to induce the lysogenic cycle where the phase DNA that we had, it gets integrated, right? It gets into the chromosomal DNA, right? This one is what? This is bacterial chromosomal DNA, right? So this bacterial chromosomal DNA that is going to integrate with the virus DNA. This blue color part is virus DNA, so it is going to get integrated into that. And because of that, and it is called prophase. Okay, it is different from the cell cycle. Cell cycle you have P, right? That is called prophase, right? So this one is PHAGE, prophase. So don't confuse there. So once the prophase is formed, this prophase continues. So here what happens, the bacteria is going to live normally and it is going to divide normally. It is going to reproduce every 20 minutes, and it makes copies of the prophase. So it keeps on copying the prophase particle and it keep on transferring to the daughter cells. Now the process continues, and sometimes what happens is these prophase, it is going to exit this lysogenic cycle and it can enter the lytic cycle. That means here we have so many host cells, and each of the host cell now can produce large number of phase or large number of viruses, and they can continue the infection at a larger scale. Now where the virus enters lytic or lysogenic, it all depends on various factors that you will definitely learn in higher classes, not here. Now I have taken another image of the human immunodeficiency virus or the HIV virus that is going to cause the AIDS that we know about. So what happens here is, as you can see here, you have the HIV virus here, and the same mechanism, HIV virus comes to the cell, right? That is the host immune cell here, right? We all know that the HIV virus, it is going to target or it is going to affect immune cells. Now what are the immune cells, right? Why it happens, which cell does it infect first, we all will learn about that in class 12th. So the moment it is going to come and attaches itself to the surface of the host cell, it is finally going to release to the DNA, right? Viral, actually in this case what you have, viral RNA. HIV is going to be a retrovirus. So this is called a retrovirus, and retrovirus typically have RNA genome. So the RNA that is there, it is going to be released, and this RNA, by a process called reverse transcription, you will learn again in higher class, it produces the DNA, right? It is going to produce the viral DNA. Now that DNA is going to integrate into the host DNA. That is, it is going to force the host cell to produce new viral RNA and the protein, and that is going to produce a large number of mature virus. And here what happens, this host cell, remember the host cell, it acts as a HIV factory, and it keeps on producing the new HIV particles. I hope you are clear on these things. Dear students, are you able to
Yes, sir.
understand this?
Yes, sir.
Okay, I'm moving on.
Yes, sir.
Good, good.
Yes, sir.
So next thing we are going to have is a viroid, right? It is not virus, it is definitely different from it. How it is different? It is simply naked piece of RNA. It is simply the RNA. Now this RNA, single stranded RNA, can be a rod type or can be branched, as you can see. There is no protein code, and simply this RNA is going to form these structures because of what? Complementary base pairing. So what do you mean by complementary base pairing? Let's say your DNA or RNA is made up of nitrogen bases. And what is nitrogen base? These are called adenine, thymine, cytosine, and guanine. Okay.
Guanine.
Yes, very good. So in simple, we can write A, T, C, G. So DNA is made up of these four components. Again, you will learn in detail later on, but for now, understand these. So always what happens, we are going to have the adenine. It is going to form hydrogen bond, double hydrogen bond with thymine, and C, the cytosine triple hydrogen bonds with guanine. So this is called base pairing. Okay, this is called base pairing. Now, this base pairing is going to allow the viroid, which is nothing but a naked RNA or a regular RNA, to form the structure that I'm talking about here. As you can see, these are the structures. Looks double stranded, but they're all, RNA molecule. They are forming this structure because of base pairing. Now, it was discovered by Danier in the year 1971, and what it is it is an infectious agent consisting only of RNA, ribonucleic acid, and there is no protective protein coat. That one difference you have to understand. Now, they typically infect the plants. There is no infection to the animal, so it is a very unique thing that you have to see, that it is affecting only the plants. For example, it causes the potato spindle tuber disease. It is also going to cause the coconut cadang disease, right? So only infection to the plants and not to the animals or humans. Now, it is having a low molecular weight compared to the viruses, low molecular weight genome or RNA. Why? Because the viral genome is single-stranded circle of RNA, as we talked about, and only it is going to have 250 to 400 base pairs, not more than that. Please switch off your microphones. Okay, so that's all regarding the viroids. Now, prions, right? Now the prions, see, there are two ways of pronunciating it. UK and US English both are different. Some of them say it is prions, some of them say it is prions. Okay, the way you want to pronounce it, well and good, no problem at all. So I would call it the prions. Now here what happens is these are proteinaceous infectious particle. So in the previous case, when we talked about the viroid, we said they are infectious particle, but they are made up of RNA based infectious agent. That is your viroid. But when we talk about prions, you have to understand that these prions are proteinaceous infectious particle. And these prions are bits of misfolded protein that have ability to spread by making other proteins misfold. So what you have to understand is we have the let's say the DNA. The DNA give rise to the RNA, and the RNA give rise to the protein. Now this protein undergoes a process called folding with the help of certain proteins like chaperones and chaperonins to produce a proper functional protein, right? It is a functional protein. Now what happens is, in this case, it is not properly folded. This folding process do not occur properly, and this produces a misfolded protein, and this misfolded protein has the ability to actually infect other organisms. So as you have seen, these are also going to have molecular weight similar to the other viruses, and because this is like the BSE or bovine spongiform encephalopathy, that is commonly known as a mad cow disease in cattle, and the similar disease in humans caused by the same prions, it is called, right, CJD or CR Jacob disease or Creutzfeldt Jacob disease. Now, what is one of the major problem with this disease or major symptom is there is rapid mental deterioration, right? That is called your, the memory is lost, right? So mental effect, the person or the individual is going to be affected. They will lose the memory, right? There is going to be rapid mental deterioration if they are infected by this particular prions. Now, the last one we're coming is going to be about the lichens. We have already learned about this in the previous case. Again, we're going to continue. So here you can see three different types of lichens. Lichens are beautiful in the nature, and there are different types also, but all are they are going to be consisting of the algae and fungi. There are only two, algae and fungi, and this is a mutualism. Right? Why I say mutualism? Because here both algae benefits and the fungi also benefits. Both algae and fungi benefits, and that's why the term mutualism, right, or mutualistic interaction. So here we say this is a symbiotic association. So here there are two components, as I said. So there is going to be a phycobiont, that is the algal component, and the mycobiont. Mycobiont means the sorry, this is fungal. Okay, the fungal component. So you have an algae and you have a fungi combined together. So what is the role of each? So the phycobiont or the fungi, the algae here, it is going to prepare the food for the fungi. You remember, the fungi is going to be heterotroph. They do not have chlorophyll. So the phycobiont or the algae prepare the food. Mycobiont or the fungi, what they do? They provide shelter, right? They provide the house, and they also absorb the mineral, nutrition, and the water and provide it to the microbiome so that it can prepare the food and give it back to the fungi.
पेपर ने रख दो
Rashmi,
पेपर
switch your microphone, please. So the lichens also act as a pollution indicator. Why we say pollution indicator? Because in the area where there is going to be pollution, or in polluted area, there is going to be no growth of the lichens. Lichens will grow only when there is less pollution or in a pollution-free environment. So that is why you have to understand that in many cases, right, there is a mechanism. When in class 12 we learn, it is on something called industrial mineralism, right? So the growth of the lichens are affected heavily by the amount of pollutants in the air, especially the air pollutants. Now, when we talk about the structure, you already have seen this earlier, but again, just I'm going to give you a brief overview. So in the center, you see us outside, you are going to have the cortex, some of the cells, a tissue, they're going to form this. And here in this area, this is going to be the algal zone. Algae are going to be there. This is going to be the definitely the green component, that is you're going to be, they are going to be containing the chlorophyll components. And here you're going to have the fungal zone. Fungi is going to be there. And here you are going to have the structure that is going to help in absorption of the nutrition and the what you call water. So that is regarding the lichens. So with this, we are coming to an end of this particular chapter. Dear students, I hope you are clear on these parts, and if you are having any doubts on what I taught to you today, please do ask now.